Welcome back. This is the second part of our Chapter 20 video, and today we're going to be focusing on capillary exchange. So the whole point of the capillaries is to let your body exchange substances, that can be nutrients, that can be gases, etc., between the blood and the tissues. When we say tissues, we just mean all the cells in your body that are not inside the blood vessels. And remember, at the very end of the first lecture, we talked about how blood flow through the capillaries needs to be slow in order to allow time for this change to happen, or for this exchange to happen. There are three main ways that substances can cross the capillary wall. You know about diffusion, that's just movement from high concentration to low concentration. Vesicular transport is things like endocytosis, exocytosis, and then finally bulk flow. Most things move by bulk flow, and that's going to be where we spend our time today. So in your book, there's a little discussion about diffusion and what types of molecules can diffuse, again, across the capillary wall. Now that can be from the blood into the tissue or from the tissue back into the blood. So another independent adventure, go check this out if you need more help with diffusion. That's in section 4.3a. The same thing with vesicular transport. So you want to understand what do capillary endothelial cells, what kinds of materials can they take in by vesicular transport, and give some examples of substances that are moved by vesicular transport. Again, if you need more review, check back to section 4.3. So we want to talk about bulk flow today. And bulk flow is the movement of large amounts of fluid and the things that are dissolved in those fluids in one direction down a pressure gradient. So we're going to move a bunch of plasma, really water, with some things dissolved in it. All that stuff together is going to cross the plasma membrane. So pressure here is a couple things. So we'll talk through kind of two different kinds of pressure today. So hydrostatic pressure, which is essentially a liquid pushing on its container. So an example of that would be blood inside the vessels, pushes on the walls of the vessels. And we're going to talk about one we know, colloid osmotic pressure. And those two things are going to determine how bulk flow happens at different parts of the capillary bed. So we use two different terms to describe the direction of bulk flow. Filtration is bulk flow out of the vessels. And where does this stuff go when it goes out of the vessels? So it's going to go into the interstitial space or interstitial fluid, which is just the fluid around cells, and then cells from there can get what they need out of that fluid. Okay, so filtration, bulk flow out of the blood vessel. This happens at the arterial end of the capillary bed. So if you remember our capillary bed, we had an artery bring blood in and a vein take blood out. At the side closest to the artery, we'll have filtration. And then reabsorption is bulk flow into blood vessels. Okay, so that we're going to take things that were in the interstitial space, they're going to cross the wall of the capillary and get back into the capillary. This is going to happen at the venous end of the capillary bed. So how can we do filtration on one side of a capillary and then reabsorption on the other? And remember, capillaries are super small. Before we can talk about how that works, we need to talk about a couple of different pressures. So hydrostatic pressure is the pressure exerted by a fluid basically on the container it's in. For this situation, we are thinking about the hydrostatic pressure of the blood. Okay, so we would say hydrostatic pressure and then we give it a little subscript B, so that's hydrostatic pressure of the blood. So let's think about this. So here's a vessel wall. Here's a capillary. Blood's going this way. Okay. That blood is a liquid and it exerts force on these vessels. So it is pushing like this on the wall of the vessels from the inside. So this is our hydrostatic pressure of the blood. Now, if our hydrostatic pressure of the blood is pushing on the wall of the vessel out, do you think that will promote filtration or reabsorption? So go answer the question and I'll wait for you here. So welcome back. Hydrostatic pressure of the blood is again force on the vessel walls. That is going to promote filtration. And again, filtration is going to be bulk flow. So we'll say fluids, bulk flow from the capillary bed, or from the inside of the capillary out to the interstitial space. Okay. So hydrostatic pressure of the blood is the main hydrostatic pressure we worry about here. And again, the effect of that hydrostatic pressure of the blood is to push materials out of the blood. Hydrostatic pressure is highest at the arterial end of the capillary. And it's going to slow down, reduce as blood moves toward the venous end. So I kind of think about it like this. So here's my blood vessel. Over here, we're at the arterial end. So here goes my blood through my capillary. Some gas exchange happens in here. Now we're over at the venous end, okay? So right here, let's do green. We got really big, I'm just going to draw a big fat arrow. Lots and lots of hydrostatic pressure. Starts to decrease, and it's small by the time we get to that venous end of the capillary bed. So this green is the hydrostatic pressure of the blood. Now, the interstitial fluid is going to be out here around the blood vessel. It exerts a little bit of hydrostatic pressure this direction, but it's so small that we just consider it zero. Okay, so our main hydrostatic pressure we're thinking about is the hydrostatic pressure of the blood pushing on the wall of its arteries, or pushing on the wall of the capillary, which encourages bulk flow out of the blood vessel. All right, our other pressure is colloid osmotic pressure. And this is the pull of water into an area by osmosis due to a higher concentration of solutes. So if you'll remember, water wants to move toward higher solutes or higher amounts of things dissolved in water. Okay. For this situation, the solutes we really care about are the proteins, especially one certain protein. So let's stop and take a break and see if you remember which protein is really important in the blood for colloid osmotic pressure. So albumin, which is that protein that's made by the liver, is our most important plasma protein for colloid osmotic pressure. So colloid osmotic pressure works because proteins are stuck in the capillary. The proteins are too big to cross this capillary wall. So let's put some proteins in here. Here's our proteins. We'll call them P's. Okay. And then there's all kinds of water in here. Okay. Water can move, proteins are stuck. Now out here in the interstitial space, there's some protein. But there's a lot more inside this blood vessel. So what that means is water is attracted to this area of higher solutes and wants to move back in here. I think I got a little bit ahead of myself, but it never hurts to say things over and over again. So the colloid osmotic pressure of the blood is going to draw fluid back into the blood, okay? Because those proteins, again, inside the blood vessel, attract water. And when water moves in this scenario, it's going to bring its solutes like ions and some nutrients with it. So, fluid is moving back into the blood. You stop and tell me, is that filtration or reabsorption? So colloid osmotic pressure encourages reabsorption, which is drawing fluid and its dissolved solutes back into the blood. Now, the interstitial fluid does exert some colloid osmotic pressure because there are some proteins there, but there are lots more proteins in the blood. So just like the interstitial hydrostatic pressure, we don't really care about the interstitial colloid osmotic pressure. So the balance or the difference between the hydrostatic pressure of the blood and the colloid osmotic pressure of the blood is going to determine whether we have filtration or reabsorption. Okay. So we use this thing net filtration pressure, which is just what's the hydrostatic pressure minus the colloid osmotic pressure. Okay. So for our purposes, we think about the hydrostatic pressure of the blood minus the colloid osmotic pressure of the blood. This is a really complicated way of saying if hydrostatic pressure is greater, right, then there'll be filtration. If colloid osmotic pressure is greater, then there'll be reabsorption. So it's really just a way of, an official way of determining which pressure is greater, but it's not hard to tell. So if net filtration pressure is positive, and remember our equation for net filtration pressure was hydrostatic pressure of the blood minus the colloid osmotic pressure of the blood. So if net filtration pressure is positive, that means this hydrostatic pressure of the blood is greater than colloid osmotic pressure. And hydrostatic pressure causes filtration, okay, or it pushes things out of the capillary. If net filtration pressure is negative, now we know that colloid osmotic pressure is bigger. That's going to cause reabsorption or bulk flow back into the capillary bed. Okay, so net filtration pressure, again, determines which direction, and by direction we mean into the capillary or out, bulk flow is going to occur. So let's draw again. Let's pretend here's our capillary bed. Okay. Blood, here's the arterial end. Somewhere in here we turn to purple. Now we're blue, there's the venous end. So over here at the arterial end, our hydrostatic pressure is very, very strong because we're very close to the heart. So the contractions of the heart are causing that blood to flow with a lot of pressure. Our colloid osmotic pressure then is pushing things back in, but it's something like this. So the hydrostatic pressure is greater, the force pushing out is greater. The direction of bulk flow will be out of the capillary. And this is filtration. So what's happening here again? Water and a bunch of dissolved solutes like ions, nutrients, not really proteins, leave the capillary bed, go out the interstitial space where they can then exchange with cells. Now, let's draw our capillary again. Here goes our blood, arterial blood, exchanging venous blood. As blood, and again, this will be our hydrostatic pressure. As blood flows through the capillary, the hydrostatic pressure decreases pretty fast. So it's going to go from here. By the time we get out here, it's teensy. Okay? That's because, again, remember the capillaries are those tiny little winding roads. So moving through all those tiny little roads basically provides friction, slows things down, decreases pressure. Okay. Colon osmotic pressure, however, stays pretty much the same as we move across. So that by the time we get over here, now our hydrostatic pressure is smaller than our colon osmotic pressure. So now our big pressure is pulling this fluid back into the blood. Okay. So direction of that blood flow or bulk flow, will be into the capillary. And we will call that what? So let's take a break and answer that question. So in summary, at the arterial end of the capillary bed, so blood's closer to the heart, the hydrostatic pressure is greater than the colon osmotic pressure. And fluid is going to move out of the blood over into the interstitial space where it can interact with the cells. That again is called filtration. By the time we get out here at the venous end, the hydrostatic pressure has decreased enough that it's smaller than the colon osmotic pressure. So now we're going to pull fluid into the blood, and that is reabsorption. Now reabsorption is really important because we don't want to be leaving a bunch of fluid out here in the interstitial space. So what would happen? We would end up with no fluid left in our blood vessels, and we'd be swelled up like the marshmallow man. Okay. So basically, again, what happens at the capillary bed? We push a bunch of fluid out, it exchanges with the cells, and then it gets drawn back into the blood vessel, back in the bloodstream, back around to the heart. So, write a paragraph, maybe, but think through how does this change in net filtration pressure make the capillary work? We just talked about it a couple different ways on a couple different slides, but think about, can you explain that in a sentence? So the role of the lymphatic system, which you're doing your independent study on of chapter 21, is also really important here because the capillary only reabsorbs about 85% of that filter fluid. So say, and this I'm making up this number, say we pushed out 10 milliliters of fluid. You would only reabsorb about 8 milliliters. So you're going to leave a certain amount of it in the interstitial space. And the lymphatic system, one of its important jobs is it returns this fluid to the blood vessels. So what would happen if the lymphatic drainage stopped? So if lymphatic drainage stopped, you would have fluid in the interstitial space. Too much, so you would swell. You would also have a low blood pressure because of a low blood volume. Because if all your plasma is in the interstitial space, it's not in your blood vessels. But again, you'll learn much more about that when you do your little study of the lymphatic system. Okay, so that's the end of this second lecture. In our blood vessel series, it was a little bit short. So again, but I really want you to think through, can you explain to me why we have filtration at the arterial end and reabsorption at the venous end?